Assalamualaikum and a very good afternoon to my supervisor, Professor Datuk Dr. Muhammad Tajudin bin Abdullah, and my co-supervisor, Associate Professor Dr. Nobuyuki Yamaguchi, and all my teammates. Okay, first of all, this is my project which is titled Arthropods cessation during decompositions of bats carcasses. Okay, for the introduction part, the decomposition process is a chemical breakdown of organic matter into its constituent by the action of decomposer. And this process have 5 stages, which is fresh, bloated, active decay, advanced decay, and dry remain. And the third one is arthropods such as spider, insect, and other are the one of the decomposer that help to maintain and balancing the ecosystem, also act as tools to determine time elapsed since death. And the last one is arthropod cessation started when the sample are dead and small insects start to appear. Okay, this is my problem statement, which is the study of the arthropod cessation of bat carcasses are still lacking of information, and we need further study of the arthropod that can be used to predict the time. And lastly is forensic entomology information is useful to assist crime investigation by the law enforcement agency. Okay, this is my objective, which is to investigate the arthropod cessation during decompositions of bat carcasses in 3 different locations in University Malaysia Terengganu. And the second one is to determine the diversity of arthropod at different decomposition stages. Okay, this is my sample site. Actually, I'm doing at 2 different locations, which is first at University Malaysia Terengganu and the Salt Waterfall Tasik Kenyir. This is the location at the UMT. 3 different locations, and then this is my methodology. First of all, I will collect the bats were collected at the island and 3 sample, and then the sample will were put in the gas trap, and I will put in the 3 different locations in the University Malaysia Terengganu, and then the sample will be observed 3 times per day, which is 8 to 10, 12 to 2, and 4 to 6. And the temperature and humidity also will be taken using the 3-in-1 anemometer, and also the insect were collected using forceps and put it in the centrifuge that contain 70% of ethanol, and then I bring the insect into the laboratory for the observation under density microscope, and will follow the Nazmi et al.'s books. Okay. This is my preliminary result. As we can see, the table 1 show the number of species and individual of insect that attract to the carcass at 3 different experimental sites. Okay, they have 4 order, 5 family, and 7 genus, which is at the different location, which is open space, shaded, and moderate sunlight. And the highest individual at moderate sunlight, and the second one is shaded area, and the least one at the open space. This is table 2 that show the arthropod succession during different decomposition stages, which is we have 3 different location, and we have different stages at each location, and we have that P is present to the present, and the dash is not present. Okay, so you can see that. Okay, this is the environment factor that we have. This is the relative humidity, and the second one is a temperature. Okay, the experiment one is referred to the empty, the blue color, and the experimental is a green color, which is at the Salt Waterfall. So yeah, and the second part is at the direct sunlight, which is same as the first graph, which figure, which is experiment 1 and experiment 2. Okay, this is the temperature and the humidity. This is only one because we at the shaded area I already done at the south only. At the UMT we not much have time sample. This is the picture of the sample that carcass from the day 1 until day 6 at the Salt Waterfall. So yes, Tasik Kenyir. Okay, from the fresh, bloated, active stage, advanced, and dry remain. This is at the open space. Same goes